right, we'll get started. How's everybody?
Good.
Yeah, good enough. So we're going to get into endocrine signaling, hormone signaling, classic hormones. We've been talking about activators, promoters, or inhibitors of cell signaling, in particular mTOR, but also MAPK, cell signaling in general. And when we look at the immune system and all of these proteins, sort of chemicals, proteins, and that really is the same thing as hormones to a degree. The classic definition of hormones is this signaling molecule released from a gland, enters circulation and binds to some target receptor somewhere, either within the cell if it's a steroid hormone, or on a cell surface receptor if it's a protein, polypeptide, amine hormone, in that protein class of hormones. So there's a lot of overlap between, say, cytokines, myokines, cytokines, and hormones. Tons of overlap, because cells just release this stuff. Cells release things. It doesn't have to be a gland, an anatomical structure designed specifically for the purpose of secreting some particular hormone. Stuff secretes it. So this diagram should make sense to you. You should pretty much know everything on here. So insulin, for example, will bind to an insulin receptor. And this could be IGF. This could be a number of ligands binding to a cell surface receptor. Here is the membrane, the phospholipid bilayer, the membrane. So we're outside of the cell, we're inside of the cell, and you get something to bind. Steps happen, but then you get to PI3K, phosphatidylinositol 3 kinase. It's a kinase. What is it kinase? What does it phosphorylate? Well, PIP2, make it into PIP3 so that PDK can activate PKB. There's also an indirect activation of, that's mTOR complex 2. I can barely read it. I doubt you can, if your eyes, or if you have like Superman eyes or something, maybe you can see it. But that's also upstream from PKB. Phosphatase intensive homolog. This is just a thing, a phosphatase that converts PIP3 back into PIP2. So there's the receptor, there's PI3K, there's PDK, PIP3, P10, and there's mTOR complex 2, there's PKB. These two we haven't talked about, nor will we talk about them, but they're phosphatases right here, and you can tell what they're phosphatasing, what phosphate they're removing. Where mTOR complex 2 is activating PKB, this thing is deactivating. Where PDK is activating AKT or PKB, this thing is deactivating. So there's just phosphatases. We have regulation, we have negative regulation all over the place. And then PKB, it's inhibiting AS160, AKT substrate of 160 kilodaltons. And this thing is a negative regulator of GLUT4 translocation. So mobilizing our GLUT4 to bring glucose into the cell. And then over here, it's inhibiting FOXO. There's glycogen synthase kinase 3, which is inhibiting glycogen synthase. This also has other mTOR regulation reactions. And then PKB to tuberin, tuberin to Reb to mTOR, mTOR to P70S6K. So all of this stuff, other than these couple of phosphatases we've talked about, as these diagrams start to make more and more appearances and become more and more cluttered, you get what's in the clutter. Like people, what my office used to look like before we went into quarantine. My office, I cleaned it up during quarantine. Like, I just gotta get rid of a bunch of stuff. But I just had piles of papers. Some of them were student papers and student exams, things like that. Some of them were research studies, all these studies that are here. I had a lot of those printed out, but I had just piles of paper like as tall as me in there, and I knew the order of them. It was cluttered, but the clutter made sense to me. It was a very organized clutter for my sake. These diagrams are similar. They seem so cluttered, but there's an elegance in their order. They're cluttered in a very specific way that cell signaling cascades can navigate. It just looks like a messy roadmap, but it's sort of not. There's an elegance to it. Okay, our team scores. And you know where to find everything. This is coming soon, I mentioned. Not this particular Evolve Your Pokemon or the BFG, but there's a growth one coming soon. It'll be posted before Friday, and you'll have a week to do it with your teens. Make sure you do this. Make sure you stratify their risk, their categorical risk, and then within those categories, make sure you do the number of risk factors. From there, it's growth, it's mTOR, it's MEPK, it's avoiding AMPK, it's how do we elicit anabolism, right? Protein turnover, that balance between catabolism, breakdown, and anabolism build up. Where is that balance? How can we lean in one direction? Remember, if you eliminate one of those, that's a disease state and it doesn't end well. If you have a pathological elimination of catabolism or promotion of mTOR, or a pathological inhibition of mTOR, both of those will end in some amount of time, some hasty period of time, in death. But what we can do is take advantage of this map, of this mTOR cell signaling map, the on-ramps and the exits, and prescribe supplements and diet and exercises and behaviors, whether it's sleep or cardiovascular endurance exercise. Prescribe things in a way that maximize traffic going to translation, right? Translation, that's hypertrophy. Translation is hypertrophy. Now, there's all these like creams and stuff where everyone's like, oh yeah, but it has collagen in my cream and stuff like that. And there's a lot of proteins, but the way we get proteins in the body is genetic expression. You don't take proteins, whole proteins, and just absorb them into your skin and incorporate them into that tissue there. The way we get proteins within our tissues is through translation. We have to have the appropriate amino acids on board. So we have to consume the right amino acids or be degrading other proteins to create a reservoir of those amino acids. And we link those amino acids together to make the appropriate proteins. Now that's the reason you can't eat insulin, right? Insulin, you can't just eat it because you break it down and then you have all those amino acids. It's just like eating, chicken nuggets or something. Same thing with growth hormone. You can't eat that. That's not a supplement that you can ingest. And so a lot of these products that you'll see, it's not quite how translation works, how hypertrophy works. Now, something like androgen creams actually do work. You can make those work. There's an absorption of them that can be effective. But for the most part, what we're looking at is transcription, translation. That's hypertrophy if you lean it in the right direction. The two mTOR complexes. There's complex 2, these diverse functions that it carries out. Complex 1 is about protein metabolism. Complex 1 is about protein turnover. It's about balancing anabolism and catabolism. And if you're favoring mTOR complex 1, promoting its activation, turning it on, you are in anabolic mode. If you're turning it off, you're in a state of catabolism. And again, it's not an all-out, like, okay, I'm just gonna go all in, it's on all the time, or it's always off. What we do is favor it or suppress it a little bit. We're not absolutists with this. But looking at, again, the broader picture, we're gonna get into amino acids. Leucine and arginine especially, these are the two most important ones that mTOR is recognizing most abundantly. Leucine seems to be the most recognized in terms of measuring phosphorylation states. Leucine is your number one, and arginine is right there behind it. Lysine also helps. There's some other amino acids we'll talk about in the nutritional section that are also of value that mTOR is recognizing. You do recognize arginine in the lysosome. Not just out here in the cytoplasm, but in the lysosome itself, you're recognizing arginine. But these signaling cascades, AMPK, you know what that is. So if there's energy stress, you're getting LKB1, liver kinase B1, promoting AMPK. AMPK is inhibiting mTOR both by turning on tuberin and, it doesn't show raptor here, but inhibition at raptor. There's MAPK signaling. There's TNF, tumor necrosis factor alpha, right there. There's the Wnt proteins. And so all this stuff is starting, should be starting to ring bells that sound familiar, ring a familiar melody on those bells. You know the complex and these things, right? So the immediate upstream and downstream stuff. Right here, this is really promotion of protein translation. Right here, this is your inhibition that you're inhibiting. It's a double negative, so there's promotion there as well. If you introduce rapamycin, you're really knocking out this one, and it's not quite as potent of an inhibitor over here, but rapamycin is extremely potent mTOR complex 1 inhibitor. Remember these cascades, mTOR and MAPK, they intersect, they communicate with each other. And it seems to be, remember, this is our mTOR scoreboard up here, and I gave it 12 points. And there's the MAPK cascade, that team, I gave 7 points. Both are very important. Here's MAPK getting to RSK, and RSK interacts with tuberin, RSK interacts with the raptor. They have shared downstream targets with mTOR and PKB, these downstream targets. And so they have intersections along the way, and they converge toward the end. Now remember, especially when we start talking about hormones, a lot of hormones activate both. A lot of hormones, we're looking at a simultaneous activation of mTOR, this cascade, and totally separately MAPK, this cascade. A lot of these hormones we're looking at are activating both, but they tend to favor PI3K, PKB, mTOR more than MAPK. Both get activated, but they're not equal in terms of their contribution to protein accretion. A huge activation of PI3K and protein accretion according to that activation, and a little activation over here and a little bit of protein translation over here. Also, it doesn't mean because both of these are active that they're equal, right? The Grammy goes to PI3K in a lot of these signaling cascades. And you don't need to know circumstances in which this cross-inhibition presents itself, or cross-activation. You don't need to know any circumstances in which that arises. Just know that it is circumstantial. And a lot of the studies are in cancer cells, because if you knock out anabolism, you're treating cancer. This lack of regulation of cell growth, that really is cancer. This dysregulated reproduction of a living thing, that's cancer. And if you can target it through the cell signaling cascades, you can treat, you can counteract the growth of those tumors, the growth and spreading metastases of cancer. And so a lot of the studies are not aimed at getting big, right? It's not, can the bodybuilder get bigger biceps? That's not the study usually. Usually the research is looking into cancer, oncology journals and with oncology applications. There are also a ton of studies on athleticism, skeletal muscle hypertrophy, but we have to take these together. We have to take these findings together, the rodents, the cancer cells, the athletes, take all those together to find the appropriate applications of mTOR and how you can intervene. But sometimes you get cross-inhibition, sometimes you get cross-activation, and they always converge. This is just an example of if you suppress one in some situations, you suppress the other. if you suppress MAPK, simultaneously you suppress mTOR. Remember, because MAPK is going to crosstalk with Raptor. MAPK is going to crosstalk with tuberin. And so if you suppress one, you suppress the crosstalk. And in other populations, other studies, other cancer lines, what you're seeing is suppress one and you compensate with the other. And so I don't think anybody has quite worked out the exact circumstances in an understandable prescriptive way where we can cover all populations and, oh, we're going to inhibit over here and we know what's going to happen over there. Research isn't quite there yet, but it's coming along and we've seen both outcomes. We've seen inhibition of one upregulates the other and inhibition of one downregulates the other. We've seen both of those. Okay, so the big picture stuff, the immune, we'll just go through it really quickly, chemical and immune. Remember a myokine, tons of these things, just proteins. These are proteins that muscle releases. Go exercise and muscles release more than lactate. Go exercise and you're generating more than hydrogen ions. You are producing a chemical environment with exercise that is diverse and not particularly well understood. And you're looking at over 600 myokines. People don't really know what they're doing. Not all of them are going to regulate hypertrophy, protein metabolism. Some of them certainly do, and some of them are pretty well characterized. So you know these ones, we've gone over these a few times. There are some that promote anabolism, promote recuperation, recovery, regeneration, repair, remodeling, re-whatevering. Some of these myokines promote it, others inhibit, and PKB, AKT or PKB, that's the major pathway through which they're doing it. Interferon gamma, that's another one. Interferon gamma, there's PI3K, there's the receptor for interferon gamma going through PI3K. Tumor necrosis factor, there's PI3K and there's the MAPK pathway over there. The Wnt proteins going through glycogen synthase kinase 3 on its way to mTOR, to promote mTOR if you inhibit that. And then remember reactive oxygen species. A huge dose of reactive oxygen species means suppression of mTOR. If it was a giant excessive dose of free radicals, reactive oxygen species of free radicals with oxygen in them, a physiological dose of reactive oxygen species is going to promote mTOR. And then arachidonic acid prostaglandins. Prostaglandins are huge MAPK activators, and you can block that either by cortisone, cortisol. That is the steroidal anti-inflammatory, would be cortisone. It's a steroid, it's made from cholesterol. So a steroidal anti-inflammatory, cortisol, blocking PLA2, phospholipase A2, which would be the release of arachidonic acid from a phospholipid bilayer. Non-steroidal anti-inflammatories, NSAIDs, are going to block COX, cyclooxygenase, which converts arachidonic acid to prostaglandins. Prostaglandins, there's MAPK signaling. Prostaglandins will also depolarize nociceptors, so they're not comfortable. And remember, Raptor, a huge hub for the function of mTOR is Raptor, recruiting those downstream targets so that mTOR can enact its phosphorylation. So that's the immune chemical stuff. Mechanical tension, we will rehash that a little bit and then get into the hormones. So mechanical tension, remember, we have mechanoreceptors. There are proteins embedded in our cells. We have proteins that are recognizing load profiles, whatever it is you're doing, right? I'm walking right now. Every foot strike, I'm interpreting that load. You know, there's a little bit of hemolysis, like a foot strike version of it, of busting up some blood cells. Every tissue is withstanding that impact force. There are proteins that are sensing that. There are proteins that are sensing the impact of my feet. in all of your tissues. You know, it's your bones, it's in skeletal muscle, it's on all of these tissues. You have these mechanoreceptors, these proteins that are sensing the load profile, and a load profile is a full profile, right? It's application of the load. Was it fast or was it slow? It is the weight of the load. How much tension is there in total? Angles, you know, are you pronated or supinated while you're holding this thing? Are you stretched? Are you over-contracted? How long are you holding this thing? All of this information gets processed by a bunch of proteins. And right here, we have these integrins, right? You have these integrins right here that are sensing. This is a transmembrane protein. It crosses that membrane. Here's the membrane, cell membrane, phospholipid bilayer, cell membrane. You have some arachidonic acid in there. For skeletal muscle, maybe 10 or 20% of the fatty acids that live in this membrane are going to be arachidonic acid. And an integrin is going to cross that membrane, transmembrane protein, and communicate with FAK, focal adhesion kinase, and relay information about the extracellular matrix immediately outside of that cell to the interior of the cell. And there's a lot of stuff that's doing it. There are a lot of proteins that are doing it. Here's the integrins and FAK, focal adhesion kinase. You have cadherins and catenins, catina. For that cell-to-cell communication, you're always going to have, if you have multiple cells in a tissue, they're adhered to each other. They communicate with each other. Cells are not isolationists. Cells speak to one another through mechanical loads and chemical signatures, chemical release and binding. And then, you know, even like, let's get into a sarcomere. Titin within the sarcomere goes from the Z-disc to the M line. That's a mechanoreceptor that senses the stretch, that senses load profiles within a sarcomere. In these myofibrils. And there's other stuff that lives in there too. Alpha actinins. Alpha actinins, there's evidence for, we're not going to talk about it here, but there's evidence for alpha actinins being mechanotransducers of sensing, having mechanosensation of what the mechanical profile is, and transmitting that message. Now, alpha actinins, there's 4 kinds of alpha actinins, 1, 2, 3, and 4 named well. And 2 and 3, these are in skeletal muscle. 2 will live anywhere in any type of skeletal muscle, any fiber type. Type 3 are a little bit more selective in the fibers. Those are gonna live in your high threshold cells, type 2 fibers. But what those do, what alpha actinins do, is they take actin and they adhere it. They bind it to the Z-disc. But it's not just, let's hold stuff in place and do nothing else. They're multitasking, right? They're structural proteins holding things in place, holding actin in place, in this case, but they're also relaying messages about the function of the muscle and the stress, the loads that are being applied to it, relaying that message. So lots of proteins are doing this. The 3 to remember, cadherins for the intercellular load, right? Cell to cell, cadherins. Titin, the intracellular load, meaning inside of the cell. That's not a transmembrane protein. And then integrins, which are another transmembrane protein going through the extracellular space. So this would be the extracellular space. There's the membrane, the plasma membrane right there. And there's the inside of the cell. A transmembrane protein crosses that membrane, sends those messages inside. And so again, if you have a multicellular structure, a tissue with a lot of cells, you have adhesion molecules. These cells communicate with one another. They don't just float in outer space unconnected to the rest of their world. So we have these bridges between cells. That cell is communicating with that cell. What this cell does informs that cell of what that greater tissue is being subjected to. Not just like, well, I'm doing all the work over here and you're not doing anything. It knows how much work its neighbor is doing. So think about skeletal muscle function. we have size principle, orderly recruitment, or size principle, where we don't just recruit muscle, willy-nilly, right? We don't just recruit muscle in any order. There's a sequence, there's a specific sequence in which cells get activated. The low threshold stuff, the stuff you easily activate, the muscle cells that are willing to be woken up with the tiniest alarm clock, just the whispering outside of the room wakes them up. Those are the weakest fibers we have. Those are the type 1 fibers, the slowest contraction, the slowest conduction velocity, and slowest contraction velocity, the most fatigue resistant also, the most energy efficient. Those are the ones that you wake up first. That might be right here. Right next to it, you might have a high threshold fiber, a big, powerful, strong thing, a big glycolytic as opposed to lipolytic, a big carb-hungry fiber that has a faster contraction velocity, as well as a nerve having a faster conduction velocity. That fiber is going to be, it's going to fatigue really quickly, but it's going to produce an enormous amount of power. That one's hard to wake up. That one's hard to wake up. But it's not completely oblivious to what the tissue is doing. Just because you go to the gym and do a set of bicep curls and finish and put the weight down, and let's say you've used 50% of that. I mean, you've like set after set, you go through the whole workout, and I don't know, you've used 75% of the cells, 25% of the cells in your bicep, of your muscle cells in your bicep, never bothered to wake up. That wasn't an alarm loud enough to get them out of bed. They're still aware of the mechanical loads that were applied on the whole tissue. They're not necessarily going to respond, certainly not as potently as type 1s might if you're using all the type 1s. But there's communication, there's cell-to-cell communication. There are load profiles that are understood and relayed. Now, with integrins, integrin right there, this little honeycombed thing, that's the extracellular matrix, that's the space outside of the cell. There's focal adhesion kinase, right? There's focal adhesion kinase. Focal adhesion kinase, FAK, is like PKB in the sense that it just goes everywhere. This is a busy kinase, a kinase that phosphorylates stuff. FA kinase, FA, let's phosphorylate. This is a really busy one. And over here, you see MAPK, downstream from FAK, MAPK. Over here, there's PI3K, getting down to PKB. But there's also other signaling cascades it's going to go through. So going through IGF, mechanical stimulus, you're going to release more IGF, insulin-like growth factor. And you'll get some, depending on the nature of the workout, depending on, are we getting endocrine release of IGF? Well, this is really downstream from growth hormone, which we'll talk about in 10 minutes or something. Downstream from growth hormone, we're getting the liver to release endocrine insulin-like growth factor, circulating in the bloodstream. But muscle cells themselves, in the presence of load profiles, will also release IGF. And we can get autocrine signaling from IGF. Now, IGF really is working through PI3K, PKB. But there's also parallel signaling. It's sending some of its traffic, not as much, some traffic through MAPK, RSK, that pathway, and converging and intersecting. Now, mechanical stimulus, remember, diacylglycerol kinase is another pathway, DGK, diacylglycerol kinase. And this is converting your diacylglycerol. Remember this thing when we were talking about fat loss? That was a big one for fat loss. We use adipose triglyceride lipase. AMPK is going to help activate adipose triglyceride lipase. All right, let's go through lipolysis and remove the first free fatty acid. Make it a free fatty acid. The first imprisoned fatty acid becomes free. Leaves its incarceration. Now we have a diacylglycerol. We could, on a diacylglycerol, we could use hormone-sensitive lipase, or why don't we phosphorylate it, DGK, diacylglycerol kinase. Let's phosphorylate that thing. Let's get some phosphatidic acid. Phosphatidic acid binds to mTOR, activates mTOR. Very anabolic, phosphatidic acid is. There are other ways. This is phosphatidylcholine. This isn't really PLD. This isn't really as much about the mechanical loads as is DGK. And this is lysophosphatidic acid. We'll talk about that over here, the conversion to phosphatidic acid. But if you take it as a supplement, really you do this conversion to lysophosphatidic acid. We'll talk about that in the supplement section. But with mechanical loading, with exercise, mechanical loading, the actual load profile, diacylglycerol, there's a triglyceride right there. And if we have a diacylglycerol, one of those fatty acids is gone. Using diacylglycerol kinase, we can convert it to phosphatidic acid. Phosphatidic acid is a promoter of mTOR, but also phosphatidic acid can bind to FKBP12. That's the rapamycin target. And so there are other functions with mTOR in which phosphatidic acid is very anabolic. It is just phosphorylate that diacylglycerol and you have an anabolic compound in a number of ways, but the direct activation of mTOR is the most researched one of those. Now remember the stretch activated channels, getting calcium into the cell, with especially eccentric loads, eccentric loads, we are activating these stretch activated channels, which are permitting the influx of calcium. And calcium does a lot. Calmodulin, calcineurin, getting that cascade activated and regulating the behavior of your satellite cells, these muscle stem cells, but also PKB-mTOR signaling a little bit more directly, we're seeing that. So there's a calmodulin activating calcineurin. And we remember that calcineurin in part controls the satellite cells. And all you have to do is block it and you see atrophy, promote it and you see growth. So getting that calcium into the cell is another effect of mechanical loading, the mechanical load profiles that induce the influx. So with mechanical loading, there's your IGF, there's your calcineurin, and remember one other effect of calcineurin is antagonism of myostatin. Myostatin is a PKB inhibitor. So if you take rapamycin, you're really knocking out almost all signaling of mTOR. Almost all signaling of mTOR is knocked out with rapamycin. If you take Wartman, which is a PI3K inhibitor, get nutrient signaling, growth factor signaling, think like IGF, insulin, stuff like this. You're knocking this stuff out. And all of these myokines we've been talking about that are going through PI3K, PKB, if you take a PI3K inhibitor, Wartman, you're knocking out that signaling cascade. So you're knocking out repair of the tissue. You're knocking out protein synthesis. You're knocking out hypertrophy if you take Wartman. But with mechanical loading, you can still get a signal through, even in the presence of Wartman. Now, not as much of a signal because mechanical loading induces IGF. Mechanical loading, you're going to get a lot of these myokines from it. So you're going to get a lot of these, part of the signal of mechanical loading is going to be going through PI3K, PKB, mTOR. Another part of the signal is not. Another part of the signal, whether it's this calcineurin pathway, whether it's phosphatidic acid, we are bypassing some of what Wartman would knock out. But Wartman is a huge anabolic inhibitor, making it very catabolic because the pathway of protein translation that is most critical, most abundant, sort of the most prolific writer of proteins. I don't know who, Charles Dickens or something, somebody who's like a really prolific writer, just so many words in a lifetime. That's PI3K. This is very prolific with those proteins. And Wartman knocks that pathway out. It puts up cones across that road. And so this is just saying that mTOR is critical to hypertrophy, and the mechanical loads are a huge part of this pathway. So mechanical stimuli induce a robust activation of rapamycin sensitive, just complex 1, dependent signaling. Changes in P70S6K, RPS6, that end pathway downstream from mTOR, phosphorylation states are a very good way of knowing if mTOR is active. Is mTOR doing its job? Let's check phosphorylation states. Very effective way of knowing if mTOR complex 1 is in hypertrophy mode. And if it's actually functioning. You could activate mTOR all you want if you don't have the amino acids present. If mTOR isn't at the work site, it's like trying to work, but it can't because it's not at the appropriate destination within the cell. You're also not going to get activation. We'll talk about that with nutrition. But mTOR is necessary for hypertrophy, right? Now, maybe it's a third, maybe it's half of this basal protein translation. But when you start looking at hypertrophy, when you start looking at let's deposit more proteins than were there to begin with, when you start looking at performance enhancement, when you start looking at growth, mTOR becomes more and more critical. So there's a lot of pathways. This isn't everything. But stretch-activated channels, getting into calmodulin and calcineurin and stuff like that. DGK, MAPK, PI3K, all of these are activated through mechanical loading independent of chemical signals, independent of IGF, and independent of these chemicals that we talked about before, the myokines and stuff like that, independent of all of that. You're activating all of these channels. And focal adhesion kinase, that's the integrin one. Integrins are the most studied, the most understood, potentially the most important of these. But remember, other proteins do it too. Other proteins, like alpha-actinin, have evidence for cell signaling, have evidence for as mechanoreceptors. Okay, questions on mechanical or anything that came before we get into hormones?
So like FAK and integrin, FAK is a type of integrin?
It's a type of kinase, it's a protein. Kinase is an ACE, it's an enzyme, all enzymes are proteins. And so integrins are the protein itself that does the sensing, but it's a mechanoreceptor. It's sensing the information in the exterior of the cell, and it relays that message through FAK. Yeah, so FAK is, think about like softball. There's like a play out in the outfield. You're not just gonna launch the ball to home plate, unless you're like Bo Jackson or something. You're not gonna like be out in like deep center and just throw the ball to home plate, right? There's like a cutoff, in this case, woman. So this would be, FAK would be kind of the cutoff woman for the play at the plate. Now, once the cutoff person gets the ball, they could throw it to anybody. Like that's a phosphate, and FAK is like, I'm holding the phosphate. All right, who do I phosphorylate? And depends on where it is. Do I need to throw it a second all of a sudden? Do I need to throw it a third? Am I just throwing it back to the pitcher? Am I chucking it into home because someone's trying to, and so you're going to phosphorylate. FAK has a lot of phosphorylation options, but that's the kinase. FAK is the kinase that does the phosphorylating. And these cell signaling cascades, often they're just called phosphorylation cascades, because phosphorylation is such a critical way of relaying a message, of activating or deactivating the next target. And FAK is attaching phosphates to things, but a number of things. There's a number of potential targets. It gets the message from integrins and makes its executive decision of where the play is. Does that?
Yeah, that's okay. Great. Thank you.
Okay, hormones now. Okay, so we'll get into the endocrine system. We'll get into hormones. Now, remember, hormones are chemical messengers. They're just chemical messengers. They are chemicals that deliver a message. And so are neurotransmitters. Neurotransmitters, and we'll talk all about neurotransmitters when we're doing brain health, you know, after the exam. We'll start the topic of exercise and the brain. What happens to the brain? And we'll talk a lot about neurotransmitters, especially when we get into flow states. I hate that term. I hate all the terms describing this. You know, I'm in the zone or flow states. A bunch of neurotransmitters. Those are chemical messengers too, but they relay the message so tiny a distance. You know, there's a little, let's say your glutamate or you know, GABA or acetylcholine or something. The distance you cross, it's so tiny. With hormones as chemical messengers, the distance you cross tends to be bigger. You know, autocrine signaling is just the cell signaling itself. The cell releases its hormone, and the hormone binds to itself. But it has to release it because the hormone has to bind to a cell surface receptor. Remember, hormones on their own aren't doing anything. Hormones, if they're not binding, are not conducting a message. They're not relaying any sort of message. They're just sort of useless until they bind. So to get autocrine signaling, the hormone has to be released from the cell, and then it has to bind to a cell surface receptor, which initiates a signaling cascade. IGF from MGF, mechano growth factor, mechanical loaded IGF from muscle cells, it gets released from the muscle cell and binds to itself. The very crude way I described it before was if you squeeze out gas right out of your colon, right? If you just unleash a bunch of gas out of your body, nobody can smell it before it exits you. It's not, I mean, maybe it's in there like making you like stomach growl or something, but nobody, it doesn't, you know, bind to receptors and stuff. Once it's out of you, then you can smell it. That's autocrine signaling. You release a thing and then it has its effect. Paracrine signaling is like your neighbor being like, was that you? That's paracrine signaling. Endocrine signaling is a big distance. And what we think of the endocrine system, that's mostly what we're talking about. That's glands releasing a chemical messenger. Glands releasing a compound into the blood, circulates around in the blood for a while, eventually binds to a receptor. Now, within hormones, there are two major classes of these hormones, right? There are steroid hormones, which are these. Okay, here's cholesterol, chemical structure of cholesterol. There's a chemical structure of testosterone. Notice the cholesterol. There's cortisol. Notice the cholesterol. Steroid hormones are made from cholesterol. And when we look at estrogens and androgens, over here, aldosterone, that's a steroid hormone. Nobody's talking about aldosterone in human performance. It's a mineralocorticoid, right? It's released from the adrenal cortex. Same with cortisol, adrenal cortex. It's a glucocorticoid. Corticoid, cortex of the adrenal gland. So you're producing steroid hormones in the adrenal gland sitting atop the kidney, not the adrenal medulla or medulla, but in the adrenal cortex, you're producing steroid hormones. You're producing amine hormones, protein, right? Amino acid hormones in the adrenal medulla or medulla. You actually do produce, a little bit of testosterone in the adrenal glands too, but it's very little. Mostly testosterone, like ladies have it too, just not very much. I'll talk about that in a second. But this is in a cascade where dehydroepiandrosterone, that's what that one is, super weak. I mean, you can go probably get this like S-Mart. This isn't banned on any like performance. It's probably, maybe it's banned in the NCAA. I'm not sure. I'm not sure if this one is. It's very weak relative to androstenedione. This is the one that became, this was the Mark McGwire drug, right? Baseball in the era of Mark McGwire and Sammy Sosa and Barry Bonds, right? Barry Bonds, who broke the record about 3 years later or whatever. Androstenedione took all of this attention, but it's really not that strong either. It's sort of silly to take androstenedione. Technically now it's in the same class as testosterone, but like this is the potent stuff, testosterone. And dihydrotestosterone, DHT, there's a lot of, you know, if you want a huge prostate or something, go for it. But these are your androgens. Androgen, genesis. Andro means man, genesis, make. These are the ones that like make the man. Males have a lot higher of these. Females have it too. And then, right, we have the estrogens as well. And this is just differences. I realize you can't see here, I'll just say it. Males and females, what expected levels would be. So this is ages, you know, late teens to 40, what you would expect, 18 to 40. A bunch of different studies. Each one of these is a different study looking at different values, and then the mean across all studies. And the value here, it's like 8 to 30. And for females, it's like 0.06 to 1.7. And again, across all of these different studies. Males, you're looking at 15-fold or higher increase in testosterone. Prepubescent, you don't see that, but post-puberty, there's a huge difference in testosterone between males and females. But women do have a little bit. They produce in their ovaries a little bit in the adrenal glands. Guys will produce a little bit in the adrenal glands as well. But it's your hypothalamus controls most of your hormones. Now, it's not everything. It's not like the entire body is controlled by the hypothalamus, but it's a major regulatory center. So for the team, for the sixth F, right, for that team, the hypothalamus, you know, controls all those Fs, you know, foods and fluids, fight and flight and fright and sex, fornicative activities. So there's a lot of stuff that's regulated by the hypothalamus by hormones. The hypothalamus tells the pituitary what hormones to get going. And often it's not even the pituitary that's releasing the terminal hormone. So for example, the HPA axis, the hypothalamic pituitary whatever axis, the HPA, you get out of the hypothalamus, you get corticotropin releasing hormone. And then you get to the pituitary, adrenocorticotropic hormone. And then you get to the adrenal gland and you get your cortisol. So the hypothalamus tells the pituitary what to do. The pituitary tells some peripheral gland what to do. Now, the hypothalamus is a part of the brain. It's not a gland. The hypothalamus is a hunk of brain. It's behind the blood-brain barrier. It's a piece of brain. And it's like the CEO. And its general manager is the pituitary, which lives outside of the blood-brain barrier. The pituitary is a gland. And most of this goes to the anterior pituitary. So for the hypothalamic pituitary gonadal axis for ovaries and testes, you get gonadotropin releasing hormone out of the hypothalamus. Gonadotropin releasing hormone. All right, let's get to the gonads, but we got to stop by the pituitary first. Get a luteinizing hormone. Out of a luteinizing hormone, we're getting our androgens. So there's a hypothalamic pituitary gonadal axis. But we also are inhibiting, when you're getting all those androgens released, you backwardly inhibit the hypothalamus. So you don't just run this thing all the time. You have enough testosterone, let's shut the whole thing down. So you will inhibit it that way. But this is the the axes at which things come from. There's a hypothalamic pituitary thyroid axis, if you're going to get to the thyroid. So again, this is just saying that, and again, all of these articles you'll see available later today on the site. But androgens, some of them are produced in the adrenal cortex, not much. Most of it comes from the gonads. And when androgens bind, the primary function of androgens is steroidal. Steroid hormones can enter the cell. They can just go through that phospholipid bilayer. I mean, they're made of cholesterol. They sort of have a backstage pass. They're born with a backstage pass, and they can just enter the cell. So steroid hormones enter the cell, and that's where they're going to find their receptors. An androgen receptor, an estrogen receptor. These are inside of the cell. Cortisol too, inside of the cell. And then once it binds, once these steroid hormones bind, for androgens in this case, bind to an androgen receptor, it will, that hormone receptor complex undergoes a conformational change, shape change, and then it will bind to a sequence of DNA. It binds to a sequence of DNA, these hormone response elements, androgen response elements if it's an androgen, and you're regulating transcription. Remember, transcription and translation. So this is the primary function of steroid hormones. Get into the cell where you will find your androgen receptor, bind to that androgen receptor, shape change, and then bind to DNA and regulate transcription. But there's more to it. There are secondary effects of testosterone that are not genomic, that are not let's bind to DNA, that are about cell signaling cascades. Secondary effects of testosterone about cell signaling cascades. And so we see, cell membrane, cell surface receptor, there are androgen receptors that have a cell surface receptor function. And we're seeing the influx of calcium downstream, an influx of calcium into the cell. And here it's showing MAPK signaling, because that's one of the things that you'll see with androgen receptor activity is MAPK signaling. You'll also see PI3K signaling, but an influx of calcium, you'll see calcineurin activity. So testosterone is not all bind to DNA and regulate transcription. Testosterone also has very potent secondary effects, signaling mTOR and sort of parallel cell signaling cascades. Testosterone and IGF, insulin-like growth factor. Testosterone is increasing the IGF signaling as well. So it will augment the effects of IGF on tissue regulation, testosterone does. And you'll see in some work, in some of these studies, a selective augmentation, hypertrophy of higher threshold motor units. So earlier I mentioned size principle or orderly recruitment. Orderly recruitment is from 1938, and that was the first time it was identified of the order that these cells are recruited in. Size principle was handed in 1957, 19 years later in the same lab, both out of Harvard. And he assigned a neuronal component. The size of the motor nerve determines the order of recruitment. So size principle is a neuronal phenomenon. But the muscle fibers that are largest, the muscle fibers that are strongest, the muscle fibers that are most explosively athletic have larger motor nerves. Those are the high threshold ones, the hard to recruit ones. And testosterone, what we're seeing this testosterone IGF relationship, you tend to see the highest threshold ones most responsive. The larger, bigger, stronger, faster, higher threshold motor units, the fibers in that pool were more responsive to this testosterone IGF signaling. So, again, muscle IGF expression with the administration of androgens. If you administer androgens, you're getting more muscle IGF, and then that corresponds to hypertrophy of the higher threshold fibers. So here's androgens. There's the control. Here's the androgens. And looking at muscle IGF-1. IGF-1 is like the adult one. Yeah, sure there's a 2, but we're not like toddlers. So IGF-1, this is the primary regulation of skeletal muscle protein synthesis. And we're seeing more IGF with the administration of androgens, more muscle IGF. We've known this for a long time. This isn't a new finding. This study is from 1993. This one's from 2000, looking at testosterone and IGF. So it's not as though steroid hormones are exclusively steroidal effects. Steroid hormones also have secondary effects through cell signaling cascades. MAPK, PI3K, calcineurin, these cascades of protein regulation, cell signaling through androgens also. And so PKB, PKB mTOR, and MAPK, RSK. Again, I use these as synecdoches. PKB for like PI3K, PKB through tuberin into Reb and mTOR. That's what I mean by PKB. MAPK, extracellular signal regulated kinase, and RSK, ribosomal S6 kinase. That's what I mean by MAPKs, cascades, not just one little thing, but the cascades. There is a dose-dependent nature in this, and so phosphorylation of S6K1, P70SSK, S6K1, remember immediately downstream from mTOR, the thing that's going to phosphorylate ribosomal protein S6. So this one's for time. So this one's time. Where time 0 minutes, 1 minute, 5 minutes, a start in 15 minutes, 30 minutes, 60 minutes. The amount of phosphorylation of mTOR activity phosphorylating this thing, there's a time course to doing it. And then, so for MAPK, set it like there's your time course for MAPK. But so once you administer androgens, you can see the downstream effects of these things. And people haven't quite outlined the pathways to a satisfying degree. But G protein coupled receptor, that's a lot of these signaling cascades. We have these G protein coupled receptors. So there's the PI3K, PKB stuff, MAPK, and you'll get a bunch of calcium into the cell as well. One other effect that testosterone has is inhibition of LKB1. Now, LKB1, liver kinase B1, it's not about the liver. It was well studied, well characterized in the liver. Tons of work in the liver, but it's ubiquitous. And so LKB1 is a direct activator of AMPK. Once you bind AMP to AMPK, bind a couple of AMPs, we're going to talk more. We'll have a whole lecture on AMPK coming up. But bind AMP, adenosine monophosphate. If you have a lot of adenosine monophosphate, that means energy stress. AMP to ATP ratio, as you get more and more and more and more AMP and less ATP, the ratio determines what binds to AMPK. ATP is an inhibitor. AMP permits activation by LKB1, liver kinase B1. Once AMPK is on, we're shutting off mTOR. And we're turning on autophagy. FOXO, ULK1, let's turn on protein degradation. Let's induce atrophy while impairing hypertrophy. That's what AMPK is doing. Testosterone is an inhibitor of LKB1. Testosterone inhibits the thing that turns on a super catabolic enzyme. A kinase that just phosphorylates catabolic things. Super catabolic. In the arguments of what the most anabolic and catabolic enzymes are, because when it comes down to it, I kind of don't give a shit about hormones. What I care about is the proteins that are doing the work. It's cheap to talk about growth hormone. It's cheap to talk about testosterone. How do these things work? Well, they work through a bunch of proteins usually. Those proteins are doing all the heavy lifting. It's much more interesting, much more honest to talk about enzymes than it is about hormones. Hormones are famous in the news. You know, steroids and whatever, growth hormone, okay, these are famous in the news. How the body actually works in real life, in biological life, enzymes are much more interesting and much more important. And if we're going to argue what the most catabolic enzyme in the body is, AMPK is a good candidate. PKA is a good candidate too. AMPK, man, that thing is catabolic. And testosterone turns off the thing that turns it on. There's a sequence to turn it on. But the thing that directly binds to it turning it on, really the activator is LKB1 among the activators. So testosterone is inhibiting that. Interestingly, so again, testosterone inhibits LKB1, but androgens inhibit and estrogens stimulate expression of LKB1 transcripts. So as we start looking at the differences between testosterone and estrogen, estrogen is another steroid hormone made from cholesterol, right? But it has cell signaling activity too. So here's LKB1. Oh, everything has a time course. Everything is dose dependent. This one is, we're talking about a dose. And so LKB1, with more and more and more dihydrotestosterone, more and more and more and more, we're getting less and less and less and less. There's regular testosterone. As you increase the dose, you get a dose response. A larger dose corresponds to a more pronounced effect. This is an androgen receptor blocker, an inhibitor. And so if you're looking at LKB1, here's how much LKB1 active with testosterone. Testosterone and a blocker. So that really tells you it's a pretty direct effect, pretty direct. Dihydrotestosterone, and the blocker. So if you introduce an androgen and an androgen blocker, a receptor blocker, an inhibitor, you don't get the suppression of LKB1. So there's something close to a direct effect. Now, again, testosterone and estrogen both, talking about these, like a world's strongest man, people aren't lean. If you look at, like, this is from Street Fighter 2, E. Honda, he was the sumo wrestler, and like he had abs, unless he was just weirdly shaped fat or something, he had like an 8-packer. But really strong people don't have like a veiny 6-pack, right? You're gonna carry a little bit more fat. And we'll talk about this later as we get into nutrition and testosterone, things like that. But the influx of calcium, both of these cause the influx of calcium, which is very rapid. That is a very rapid effect. And so with estrogen, moving out of testosterone and into estrogen, again, the primary effects are steroidal, genomic, just like with testosterone. It is a steroid hormone made from cholesterol. It can just go into the cell. It has a backstage pass. It just goes into the cell and finds its receptors there and has genomic effects. Secondarily, it has cell signaling effects. And it's a little bit fascinating. You see, steroid hormones are slow. Steroid hormones have very slow effects. They are slow to take effect and they're long lasting. Find somebody doing anabolic androgenic steroids, with a needle full of testosterone, or Deca, or whatever it is they're doing, Winstrol or whatever. Find somebody doing steroids, and like, how often are they injecting? Like once a week, something like that. Long-lasting, slow-acting, long-lasting effects, steroid hormones. Amine, peptide, polypeptide, protein, that class of hormones, right? Those hormones that are made out of a string of amino acids, an amine hormone, one amino acid. Those hormones are fast-acting and short-lasting. Steroid hormones are slow to take effect, and that effect is slow to decay. When you start seeing rapid effects with steroid hormones, you know there's something else going on. There's something else going on when you see rapid effects. And the calcium influx is with both estrogen and testosterone. Now, what's interesting about estrogen, I call it the deptor of steroid hormones. Now, deptor is a protein, and estrogen, right? That's made from cholesterol. So the chemical structure, one has nothing to do with the other. But remember, deptor has anabolic and catabolic effects. Deptor is inhibiting catabolism while inhibiting mTOR. You know, let's turn on PKB, but let's turn off mTOR. You know, let's get in the way of autophagy while getting in the way of anabolism. So that's what deptor does. And estrogen has a sort of a similar effect, sort of pros and cons, depending on, are you looking to be anabolic or are you looking to be catabolic? So this is just showing the estrogen receptors, and we're looking at calcium, calmodulin, calcium urine signaling. There's PI3K signaling in there. And so the estrogen receptor, again, all of these available later today. The estrogen receptor belongs to a superfamily of ligand-inducible transcription factors that, upon binding ligand, undergo conformational change that facilitates their association with both DNA and co-regulatory components. So regular steroid stuff and cell signaling stuff, along with the ability to regulate gene transcriptional activity, just like we talked about with testosterone transcription. The unliganded and liganded states of the estrogen receptor also participate in complexes of proteins that serve a variety of cellular functions. Again, cell signaling. This receptor modulates cell cycle progression and alters calcium responses. So one of the things is this relationship that estrogen has with tuberin, the tuberous sclerosis complex, immediately upstream with mTOR. Tuberin can modulate, can inhibit gene expression actions of estrogen. So tuberin is sort of an antagonist of estrogen. But estrogen is an antagonist of tuberin. Estrogen phosphorylates, deactivates tuberin, just like PKB does. So estradiol rapidly increase the phosphorylation of downstream targets of mTOR. Introduce this, and mTOR is activated. Again, those are the downstream targets. Wartman, a PI3K inhibitor, and rapamycin blocked the estrogen-induced activation of P70S6K. Again, mTOR signaling with estrogen is turned on, but if you introduce Wartman, it's turned off. And it's very fast. This stuff's very fast. So this effect of going through PKB, PI3K, PKB, through estrogen by its inhibition of tuberin. The estrogen inhibits tuberin. And so this is P70S6K, phosphorylation of P70S6K, and here's a time course, right? So it just takes a little while to get going. Introduce rapamycin or Wartman, and you know, bye-bye. Goodbye signaling. Here's ribosomal protein S6. Takes a little bit of time to get going. Same thing. What we're looking at these, the 4EBP1, remember downstream target of mTOR. There's a time course for this to happen. The steroidal time course is slow. The time course of cell signaling, this is a little bit faster. So tuberin, estrogen is going to turn off. But remember, estrogen promotes LKB1. LKB1 turns on AMPK. Estrogen both directly and indirectly activates AMPK. And AMPK is super, super, super catabolic. So estrogen, I would argue, is the deptor of steroid hormones. It's anabolic on one hand, it's catabolic on the other. Now we know all about insulin and IGF, PI3K signaling. We've gone over that signaling a lot, like PI3K, PKB. That's insulin and IGF. Now, IGF, you're going to see some parallel MAPK over there. IGF does a little bit of that, but remember, IGF is mostly going to be PI3K, of how IGF, insulin-like growth factor, is enacting its anabolism. Now, growth hormone. Again, people talk about this stuff in the news, and I roll my eyes, my nose, my ear, all of my anatomy just starts rolling when I hear the way growth hormone is talked about in sports. Growth hormone is talked about on the news. Growth hormone is talked about in athletics. Most of its effect on skeletal muscle tissue remodeling is because it causes the release of IGF, of insulin-like growth factor. The hypothalamic pituitary liver axis, you're going to get growth hormone releasing hormone out of the hypothalamus. Obviously, that causes the release of growth hormone. Growth hormone releasing hormone causes the release of growth hormone out of the pituitary, the anterior pituitary. Later, that causes the liver to release IGF. IGF does a bunch of PI3K signaling and a little bit of MAPK. So growth hormone, how is this thing enacting its effects? Remember, fasting increases growth hormone, and nobody's growing during fasting. You get more growth hormone, but in fasting you suppress IGF. So it's ludicrous to say, I'm gonna eat nothing and get big. Come on, we know better. Despite elevated growth hormone. So this idea that like a growth hormone isn't even like a single thing. There are a bunch of aggregates of growth hormone. But the major tissue interaction is going to be through IGF, through insulin-like growth factor. Now, growth hormone binding to its receptor, you see JAK-STAT signaling. JAK-STAT, Janus kinase, signal transducer activator of transcription. This signaling cascade is not as well understood, but there's parallel signaling you see with NAPK and PI3K. So here's JAK-STAT. Okay, here's PI3K, PKB, mTOR. There's NAPK getting activated. There's a lot of crosstalk with growth hormone signaling. So growth hormone does do a lot of things. When we talk about hypertrophy, if growth hormone did what we say it does directly, if growth hormone were directly responsible, as opposed to having a cascade that relies on other stuff to do all the work for it, fasting would make us huge. So mTOR, this is the major signaling cascade with growth hormone, though, is through PKB, mTOR. Yes, when you look at this, JAK-STAT, MAPK, PI3K, the PI3K, PKB cascade turns out to be the major signaling cascade for growth hormone. Thyroid hormones, T3, T4, right, these thyroid hormones, triiodothyronine, thyroxine, PI3K, PKB signaling. Both of these articles will be available, but listen to this one, same thing. And looking at T4 and the placebo, and depending on where you're phosphorylating things, remember there are multiple phosphorylation sites. Here we're looking at PKB and S6K being having an elevated phosphorylation state. This one's more fascinating. Again, PKB, whatever, the S6K, but ribosomal protein S6 and S6K, downstream from mTOR. P70 S6K, ribosomal protein S6. T4, okay, huge phosphorylation state. T4 with rapamycin. T4 with rapamycin. So the anabolic signaling of thyroid hormones just gets abolished with rapamycin. Now over here, it's sort of fascinating with MAPK signaling, right here. MAPK signaling in the presence of rapamycin. There seems to be a compensatory effect. It's not a very good signaler of MAPK. Thyroid hormones. And thyroid hormones are interesting because it's an amine hormone, but it actually does get into the cell similar to a steroid hormone. But PI3K, PKB, that's really the effect of anabolism coming from the thyroid hormones with a little bit of parallel MAPK. So endocrine system, lots of things. And most of this effect endocrine system with glands releasing hormones, inducing signaling cascades. It's a bunch of PI3K, PKB for the most part. So we've done the immune system, chemicals, right? Tons of stuff. We've done mechanical loading, sort of tons of stuff. Very potent over here, sort of bypasses some of the signaling of these two. This one does. And the endocrine system is done. What we have next is nutrition. What you put in your mouth and chew and swallow, or drink or whatever. What you consume in your mouth hole is what comes next. And that's a little bit different for the function of mTOR. Okay. That was a lot of words. Are you okay? So it's a couple of thumbs up and mostly head nods in the direction of no. All right, go rest. Go rest. And so remember, I won't be here on Friday. So you can feel free to come, but you'll be coming to potentially a lonely classroom, unless everyone else shows up too. I will get the case study posted, and you have a week to do it. So it'll be due the following Friday.